Get started? All right. Good morning. So I'm going to go ahead and try to finish the epistasis directions first. I went ahead and pre-written in some of the stuff so I can go at maybe a little bit faster pace than I do when I'm teaching so I don't fall quite as far behind. Then I'll move to the evolution of metabolism section. And I've kind of blended the tutorial in with the lecture in a lot of different spots. And to do the tutorial sort of stuff involves running some code in R, as I think I mentioned yesterday. And so I sent 3 little files with R code in, and so they should be on the website, I guess, to download. Yeah, so at some point at your leisure, it'd be good to get those downloaded before we get to them. Because that'll be, like I said, kind of blended in as we get going on the metabolism. But let's go ahead and get back to epistasis and how mutations interact with each other. And I just wanted to kind of recap where we left off last time, thinking about within proteins, where this is kind of the sort of picture that, like I said, has actually been quite common now to see in different systems where the same mutation that occurred within a protein, when tested on different possible combinations of the other mutations that occurred, will often have very, very widely different effects, which you can see here on the y-axis. The magnitude of those effects are often massive compared to, say, its average effect. And with something like how fit was that allele in the first place, the impact of another allele being tested on those backgrounds is relatively idiosyncratic. There's no particular kind of scaling with the background. And those 3 parts is what we're going to mainly be comparing to, looking at epistasis between genes when a whole organism evolves. So, where was I? So I had talked about this model system that we were working on in terms of an introduced new metabolic pathway, the cells initially growing really poorly, but in a relatively short course, improving by a rather substantial amount. And so we wanted to be able to ask the same sort of question about epistasis, but kind of across the entire genome. And for this, we looked at kind of 2 types of comparisons that I'm going to bring up. One of them is combining alleles that arose in separate adaptive trajectories. So from population 1, from population 2, from population 3, what if I take a beneficial allele from population 3, combine it with one from population 1, in this case that affect the same pathway, the same kind of physiology, and if that one was good, and this one is good, if I put them both together, will it be awesome? Will it just be kind of meh, or will it even make things bad, etc.? So there's that kind of a test. And then there was a second kind of test, which is looking at, very similar to the protein work, taking a strain that has multiple different mutations across the genome and now combining those in all possible combinations. And so the first was a pairwise test across separate trajectories versus kind of the all combinations for mutations that occurred within a single trajectory. And in this case, with the single adaptive trajectory, we had, am I going to bring it up here? Yeah. No, we'll get to that when we get to there, what those mutations were. All right. Yes? I don't know. You like population size. I don't know in this case how strong of an effect I necessarily expect population size to have. It may affect things some, because again, this kind of began to come up a little bit. I know will come up more. The bigger the population size, the more other beneficial alleles you're competing against, the more kind of extremely good you have to be, as well as your progeny have to continue to be able to evolve to be. And so it may make things a little bit more extreme in terms of fitness, and it's hard to know. I think right now it'd just be guessing if you were more extreme in fitness or less extreme in fitness to be a winner, does it make your epistasis patterns stronger or different? I don't think we have any information. I don't know. Do you know? Yeah. I don't think anybody's done that kind of test. It's an interesting question. I don't think people have done that yet. Unfortunately, it's a lot of work to do even just once or a couple times. So probably as things are getting easier, we'll get more of that kind of stuff, but I haven't seen that yet. So first tackling this first, number one here, combining pairs from different populations. And so it turned out, not surprisingly, that we introduce a novel pathway and every single population gets beneficial mutations within this novel pathway that change its expression level. It's kind of guilty before the experiment even started as being a fairly likely place that something might need to change. And these occurred very, very early. There was actually a large number of different alleles even within the same pathway. Of course, they can't all win because they're at the same locus, and so there's clonal interference amongst them. That's a whole neat story too, but I'm not going to go down that road. These mutations, this was work by a former student of mine now at National Taiwan University, David Cho, and he found they kind of fell into three different classes. And interestingly, none of them were the class that you might think of. If I said a gene needs to evolve different expression level, where's a very likely way to do it? Many of you might say, well, how about a promoter mutation? Right? That seems like, and that's been very common. In the Lenski lines, that's one of the most common kinds of mutations that are just right upstream of genes and often either turning up or down expression. We saw none, no promoter mutations, and I'll be able to explain to you why by later. But we did get kind of three classes of mutations to this system. We had a number that fell between the two genes of the pathway, which was kind of interesting. They're in the pathway, if I spell pathway correctly. But odd things like changing the ATG of the second gene to something else, a non-canonical start codon, or deleting the ribosome binding site of the second gene, various other sorts of things. And so they were very particularly located, and some of them seemed to be very odd sorts of mutations to be something beneficial. So there was that kind of class. There was another class that was in plasmid replication machinery. So I just said some words that may not be familiar to you. So I put this, to make my life easy, or at least we thought it would make it easy, I put this pathway together on a plasmid, which is kind of a free-floating mini chromosome, if you want to think of it as such. And it did make it easy because we could take it out and sequence that locus back before Illumina sequencing was trivial. There was not a single evolved sequenced genome for 5 years more after I started this work, and then that style of doing stuff started to happen. And so a number of them affected plasmid replication, and then there's a third class, which unfortunately I'm going to ignore today, where there was integration the plasmid into the chromosome, into the host genome. And I'm not sure whether I'll actually end up talking about horizontal gene transfer or not given time, but this is one of the key ways that genes can end up into organisms' genomes, is that they may come in on a mobile genetic element, but then somehow get sucked in. And so it was really cool that we got this kind of event. Strangely enough, this happened many times in every single population, and in only maybe 3 of the 8, I think it went to fixation. And most of the other times it was there, they started to shoot up in frequency, and then they started to fall in frequency because other trajectories were even more fit. And so they tended to lose out due to clonal interference. Yeah? That'll be in the plasmid, yeah. Ploidy's coming, yeah. Yeah, absolutely. And so this actually does change copy number 2. So the copy number of this plasmid is about 10 normally. And so once it's into the host genome, it's 1. No, in this case, none. Yeah. It certainly has happened in bacterial systems, and it's very common in yeast systems. Yeah. In this case, we did not have any of the John Rothian stuff. Yeah. Great. So we had these different classes. This one, unfortunately, this last one that was kind of neat, is a pain in the ass to genetically make. And so for now, we're going to ignore that. But we had these other classes, and we could combine mutations. Like, what about one that affected like the genes itself and one on the plasmid? Those sorts of combinations. And so what do these mutations do? So David measured multiple different properties. He measured plasmid copy number, so getting it ploidy. And so that's the first set of the black and white heat maps for each of these different mutations. Measured the mRNA levels, measured the protein levels, et cetera. And you can see that you get kind of a different sort of pattern. For the mutations that fell between the two genes, the plasmid copy number was unchanged. Not surprisingly, they weren't in genes that affect that. But both the transcript and the enzymes, way lower, way lower. So interesting. So they affect expression and actually turn the pathway that we just introduced and the cells need down. Okay. What about the second class that has got the blue box around it? These affected plasmid copy number, you can see it on the top, was lower, and essentially, like this B5 really turned down plasmid copy number and also turned down everything else because of that. Because the ploidy went down, instead of having 10 copies, like this one I think has about 2 copies per cell, you have lower transcript, lower protein, et cetera. So all these beneficial mutations affected expression, but they did it in a very different way. One affected ploidy, the copy number of the plasmid, and the other affected expression per copy, the other class. Yeah, but in reverse, where it's bringing it down, exactly. So we either have low expression per copy or lower copy number of the plasmid, all right? So we have these two different phenotypes. So getting back to the concept then of epistasis, how might these interact, right? And so you can think of them with this little cartoon diagram. One of them decreases the expression per copy. One of them decreases the copy number. So let's imagine each one decreased by 1/3. How much do you think you'd see in the bottom corner if we did both mutations? What do you think a null prediction might be if it was like 1/3 and 1/3? So drop by 2/3 and then drop by another 2/3 to a negative 1/3. Like it's hard to be additive when each mutation in that case would be like more than a 50% change. Yeah. Do you remember the other null model I threw forward? Multiplicative. So if it was 1/3 and 1/3, perhaps it'd be like 1/9 the original, right? You could just take the two proportional changes, and if they were independent events, if this was like this and this was like that, then maybe that would look something like that, right? And indeed, this is what you see. So these two classes do look like they are kind of independent molecular events. If you take each of the two single measures and you compare it to the double measure, it predicts it really quite well, right? So this is an example of where a multiplicative model, I think, is appropriate for two independent changes to a phenotype to each other. Now, this is for expression levels. Predicts it great. That's fantastic. Well, that's nice, but I'm interested in adaptation. What about fitness? This is what that looks like. It's not as impressive, is it? Right? And it's not just that there's noise. Like there's clearly something wrong going on. Like it's under-predicting a set of mutations, and sometimes you have mutations that, when combined, are jointly deleterious. And in terms of expression level, they would have been somewhere up there. So, like, what gives? So I'm gonna leave this story for the moment as a problem, and we're going to come back to that when I talk about evolution and metabolism, because ultimately, with a very simple little algebraic model, we're going to be able to predict all of the double mutants from the properties of the singles. And the simplistic view of just, well, let's imagine that they're independent, clearly is not true. They are dependent upon each other, but I'm going to be able to explain that later, but we're going to have to build up to that. So I'm going to put this to the side for the moment and look at the other sort of combinations. So this is the mutations across the genome. And there were so after 600 generations, we saw 9 mutations. 3 were in C1 metabolism. And these were, I'll just leave it at 3 in C1 metabolism. And then there were 6 others. We do know something about some of those, but we lumped those together as one allele. And that was the one that was in red up above. So this chart is similar, so this is the same kind of experiment that Dan Weinrich did with mutations within proteins, but now we're looking at mutations across the genome. And to make sure that, I talked about this before, but just to make sure that this is clear, for these, each little box like this, these numbers, the top set of numbers represent, the pathway, a gene that's involved in interconversion of NADH and NADPH, a gene that's involved in making the cofactor of the new pathway that's in these cells, and then this, quote, genomic background. So when we say something like 0010, it would be ancestral alleles for these 3 loci, and it's just a single mutant where it's only the evolved beneficial mutation for GSHA. So that would be like this guy. And then underneath it is the fitness value. Does that make sense? All right, so then, all right, so you can look at these beautiful data. And how is this chart organized? You can see that it goes from the ancestral strain, all zeros, to the fully evolved strain, all ones, which each of these rows then represent the single mutants, the double mutants, and the triple mutants. Right? Okay, and then we can take a look at each color. So what I've done with each of the edges between these different nodes is the selective effect of that allele on that background. All right? So for the singles, it's really simple. If the fitness is 1.166, the ancestor by definition is 1. All right, that's, you get 0.166. For all the other edges, so like that green 0.406, that's taking that 1.639 and dividing it by this, because you're asking kind of compared to this background, what's the proportional improvement in fitness on that background? So it's not subtracting the 2 values, it's actually the 1.639 divided by this, because you're asking what proportional increase in fitness did that allele have? And the nice thing is I have, for example, 8 places where the green allele is added to a different background. So I can ask 8 different times, what effect did the GSHA allele have? And if there was no epistasis, it would be the same number on each one of these lines. It would be 0.3, or give or take measurement noise, right? And so you can look then, kind of for each one of these. There are a couple key patterns that are somewhat different than was seen with Dan Weinrich's data. So if you look at all the values on all the edges, what's one, like if we look at it in total, what kind of value is missing from the data set? Numerical value. Negative. They're all positive. And actually positive and non-zero even, right? So what does that mean? In words, every mutation was beneficial on every other background. Or you could say it as there was no sign epistasis. There was not a single case in all these combinations where a mutation that might have been individually good that was actually then deleterious on some other background, right? So that, I think, is the first major thing that's really clear. No sign epistasis. All alleles beneficial on all backgrounds. Okay. There's one other. So what does this tell us about? So again, it's a hokey game, but let's use it for a second. Imagine these were the only mutations you could take. You could at least ask, like, to get to that particular kind of quadruple mutant, how many of the paths would be selectively accessible that selection could drive you up? Or I could phrase it the other way, 1 minus, how many of the paths are not possible? Right? Nothing's not possible, right? There's not a single flat or downhill step. So you could drop, marbles at the top. They'll roll. It's only got one peak. They'll all roll to the top, to the evolved allele, which was also true of the Weinrich data. But in this case, they can take any possible path. All of them could be driven by selective pressures, perfectly reasonable. So every trajectory is possible. And if it's biallelic like this, there's however many alleles, it's that many factorial possible combinations up to the top. So there were 120 possibilities for the Weinrich data. 24 here. The color difference, yeah, sure, sorry. So each color, though the yellow and the red aren't showing up too well compared to each other, each color represents the same allele being looked at across this. And so, like, let's take this yellowish orange one. That's actually the mutations to the new pathway, the kind of thing that I talked about just before. And so we can look at, it had a 14% effect when introduced on the background that had no beneficial alleles already in it. We could go up to the top to that 1.812, the 0.111. And if it was like the last allele to occur, it has only a 7% effect. And you can look, so it's 7% there, 8.8, 10.5% effect, et cetera. And so if you look at one color only, you're looking at the effect of that allele on adding that to many possible backgrounds. Or you can look at the green on many possible backgrounds. You can look at the blue. So it's kind of nice. You have only, in this case, 16 fitness data points, but you can make a whole bunch of these kind of pairwise comparisons. And we'll look at them kind of all at once in just a sec. All right, so every trajectory is possible. Now, what about the actual magnitude? Anybody notice a trend? Or at least 3 of the colors. Like, look at the green values, for example. It's it's like a 51% effect here. Up at the top, it's a 35% effect. And as you march up this tree, it's kind of getting smaller and smaller and smaller in a fairly consistent sort of way. If you look at the yellow, from 14% up to 7. If you look at the red, from like 17% up to like 10, 11%. Right? And so there's a trend. We're going to look at that in just a second. But for 3 of the 4 alleles, decline in magnitude with fitness, yes.
But I would expect that it's not depending on which background I'm using the mutation, it's not the relative amount that should stay the same, it should be the absolute amount that should stay the same. That seems to hold out here.
Well, so this is what we actually talked about yesterday's lecture, right? In terms of whether a multiplicative or an additive null model is reasonable. And when I was trying to make the case, like with the enzyme expression data, that if these were independent functions for the cell, then the null expectation here, I think, would be that they would have a constant proportional effect, like the expression and copy number. If they actually were independent mutations, then it's the fold change of one times the fold change of the other. Just like separate life stages of number of progeny and like survival of the progeny. those are, if they're two separate things, then there's not going to necessarily be some constant kind of like absolute amount of fitness change, but it would actually be a proportional amount. You know, if you double survival, but you're doubling survival of what, right? It's not just like I'll always add 50 babies to any given clutch. What if you only have 10 kids, right? You know, like, you know, and so, you know, I think. So I guess there's an important point that, to make here is I think there's a distinction between what model best fits real data versus what model represents a null interaction, right? Because if you could take real data and ask what possible model does the data most look like and set that as your ground state and say, this seems to be the average of what things are. But what if, I mean, if mutations tend to be synergistic or tend to be antagonistic, you've just canceled that out and you said that that's now the new norm. And you'd only be looking at like what's more synergistic or more antagonistic compared to average. Whereas what I'm trying to ask here is, compared to independence, is the normal independence, is the normal that you get less than that, you know, et cetera. And so I feel like you know, I think it's a reasonable you know, question to have, but I think it depends upon exactly kind of what sort of answer you want to get from it. Yeah. So to be able to see what we just said about magnitude, I took all of those fitness values for any given allele, like for the green one, and I set whatever its highest value was, so in that case, like the 0.509 as like the maximum possible, and then put kind of the proportion of that maximum selective advantage for each of the different alleles, and then compared to the fitness of its background. But because all the fitnesses of the backgrounds were kind of slid all over the place, I normalized those between 0 and 1, between the least fit background, which would be the ancestor, and the most fit background that they were added to. So just kind of scale it all so it all kind of fit into a unit box. And you can see for the green, the yellow, for the red, a very clear pattern of diminishing returns. But interestingly, not for the blue mutation. The blue mutation, if you look through, there's relatively small deviances, you know, of maybe 2 to 4%, around an average of about 10%. It seems to hold pretty much constant, proportionally constant. It seems to always be, give or take, a 10% effect. So the blue guy is pretty flat, but all the others, you know, are dropping down in a fairly consistent manner, that sort of thing. And then I showed you one of the alleles from the Dan Weinreich's work. Now each color represents, you know, looking at each one of those 5 alleles that was within a protein, doing the same sort of analysis, and you can see for any of the 5 alleles that they looked at, the same story, that they always have like really pretty wide epistasis, sign epistasis, and no particular scaling with background. And so, you know, within protein, between genes, seem to have a very different kind of picture. So I would say, so for a cross genome, see what can be called diminishing returns, or you can call it antagonistic. That the epistasis tends to be fairly small compared to the average selective coefficient. And so like the green mutation has an average effect of about 40%, and the epistasis changes that by, by 5 to 10% from its average. So it's measurable, it's definitely significant, but it's fairly modest. And for all of these, the deviations from epistasis are smaller than the average effect of the mutation, whereas for the protein mutations, it is quite the opposite. The deviations can be many times larger than the average.
Do you think this is this decrease in magnitude? Mm-hmm. I think it's because of selection. It's kind of thing of the you don't want to overshoot, right?
But this is fitness. There's no intermediate optimum for fitness, right? So this is not like tail length or something like that, where I think that that would have a different kind of meaning. Here, this is growth rate. And so if anything, so I think it's actually an even stronger case. This is a biased set of mutations. I'm going to talk about bias in a couple places. These are the mutations that won, right? This was a background that did well. And if anything, if there could have been synergistic epistasis, it would have been all the better, right? There was all the bias set up in the world to have the kind of the fittest possible background to outcompete the many other beneficial trajectories that were also occurring at the same time. And so the fact that even when there's selection to have as high of fitness as possible, you still see this diminishing returns. I think it actually makes an even stronger case, so as to say, that even in light of selection for the total trait of fitness, you still don't interact particularly well with each other. Yeah. It scales generically with fitness. For 3 of the 4, certainly not universal. And it's very different than the beta-lactamase. One thing then that is important about this is a contributing factor to adaptation slowing down, which you saw from our data, from Rich Lenski's data, et cetera. And so you can imagine, like one way of thinking about it is, because of the bias, because beneficial alleles have to be escape drift and all the cool stuff that Lindy's going to talk about pretty soon, that the bigger selective effect you have, better chance you can run away from the number 0. Is going to bias towards big effect mutations. But also the big effect mutations are going to be rising, even if those that do establish will rise at faster rate than less beneficial mutations, et cetera. That you can imagine if there was a deck of cards and that represent, their kind of like their face value would represent kind of what, how beneficial they were, you would imagine adaptation might, generally first get led off by like aces and then kings and queens and jacks and eventually get down to kind of smaller effects if they had a kind of constant effect. But this is saying that there's a second thing that fine, maybe aces are the best cards to be able to collect, but the first time you get an ace, it has a certain value, but the second time you get an ace, it has less value now that you already had the first one. Like the kind of the amount of effect is actually declining as adaptation proceeds. And so there's both effects, kind of the things that are possible. There's going to be a bias towards the bigger effect things happening early, but also the stacking of alleles with each other tends to be less beneficial than if you could just add up all of their individual effects upon fitness. All right, but this is one weirdo system that some weirdo person's telling you about, where I put in some novel pathway and strange things happened. Is this likely to occur under any other circumstance? Yeah, let me actually do that, and I'm going to come back to the why. So for this, we could turn to kind of the best understood system, the Lenski lines, and you guys heard about this last week, right? That Rich, back in 1988, started a number of populations long enough ago that the world looked fairly different. So I would say, personally, I think he could fit into Bollywood just fine, don't you think? Like that looks pretty, on par still for the day. So as you guys probably already heard from this, this is in the best understood situation. We see very similar kind of fitness trajectories, fast early, slow later, but even for a substantially longer x-axis, longer than some of your lifetimes. And we see a whole bunch of genomic change during this time. I'll perhaps come back and talk a little bit about some of this. And even can, from individual isolates could begin to find out, of course, what were the beneficial mutations that caused adaptation. But so I was at a physics meeting in Aspen, because somehow I like to, I guess, pretend to be a quantitative biologist, a quantitative sort of person, and manage to be around the edge of these groups. And thankfully, physicists like evolution, and so it's been fun. there was a session on epistasis where Dan Weiner gave a talk on the stuff that I showed you, and then I gave a talk on the stuff I just showed you, and then Tim Cooper, who worked on the Lenski lines, gave a talk, and I started my talk with mentioning Dan Weinreich's work, and then here's what we saw. He started his talk mentioning Dan Weinreich's work, and then he basically presented the exact same talk I just showed you on methylobacterium. The amazingly similar stuff I'll show you in just a second. But his was with the E. coli lines, and he took, he took back at this inner line of this little mandala diagram was at 2,000 generations, and there were 1, 2, 3, 4, somewhere else, 5 mutations. He made all combos of 5 mutations from E. coli evolved on glucose. And same sort of plot that we had, just a little bit different in terms of kind of coloring choices, et cetera. 4 out of the 5 showed diminishing returns. Exact same phenomenon. So it's also, I think, really important. We know that ours can continue to adapt, but this was only 2,000 generations in. We damn well know that the Lenski lines were nowhere close to being done. So it's not like you get diminishing returns because you're almost at the fitness peak. So things just couldn't possibly get any better. No, they get better like for the next several decades of the E. coli's life. Like this is only represents the first few steps of a bug eating what its preferred resource is. Now, obviously, in a new environment, in lab is different than being in the gut of a person. But see basically identical sort of trend where the epistasis was smaller than average selective coefficients, no significant sign epistasis. And 4 out of 5 alleles with diminishing returns. So, at this meeting, thankfully, I knew Tim. We're good friends. And in fact, we were sharing a hotel room to keep costs down. And so after we each gave our talk, we went back to the room and kind of stared at each other. And we're like, so what you going to do with your paper? How soon are you going to be publishing? Should we make one big paper? What should we do? Because we clearly have the exact same tale to tell, and we're both junior faculty at the time and trying to figure out how to make this all work out. And so we decided that we would submit on the exact same day, and we wrote one cover letter to Science that all of us signed, saying, if you see something once in experimental evolution, you've at least demonstrated this is possible. This could happen. Here's an example, all right? This is at least within the realm of possibility. But if you see it twice for 2 rather different systems, it's only twice, but even still, it's beginning to give a picture. I think they really support each other that this might actually be a fairly common sort of pattern. And thankfully, that worked out. And so that, they ended up coming out kind of back to back in sort of way. And it's not to say this is the only thing that could be seen. So let me see if I jumped, so let's see if I can keep track of what I left behind. 1 year later, all right. So there definitely have been loci with synergistic effect. So 1 of the 5 from E. coli, which happened to be part of the PTS system for transport, glucose transport. Showed the opposite effect. It actually was generically, so this definitely goes against an additive model. Its proportional effect goes up on the backgrounds that it was added to. So it becomes more and more proportionally beneficial on the fitter and fitter backgrounds that it was seen on. And earlier, David Cho had seen the same sort of thing where we had one allele that happened to be a cobalt transporter. Where we saw the same thing, where it was an allele that had very little selective benefit, like only about a 3%, this is proportional, about a 3% proportional effect on a relatively slow strain, but on a faster strain had an 18% proportional effect, right? And so very strong synergy. The neat thing about what David did, so these both represent epistasis, what I've shown so far, where epistasis, we're looking at an interaction, and you could call it a, quote, G by G, the interaction of one genotype with another genotype. But David did a second experiment where he looked at what's called a genotype by environment, or G by E. Somehow the X is pronounced by, in terms of how people say that, where he simply grew the fast wild-type strain at different temperatures to speed it up or slow it down. All right, does that make sense? He took the same strain that he used with these allele comparisons in black, it's there in the circle, and then instead of just growing at 30, which all of our other data is, he turned the incubator down to 25, or to 20, or to 15 degrees Celsius to slow it down, to slow it down, to slow it down, and the allele became less and less beneficial as you slow the strain down, right? And so it's truly, it's not so much like it is, that's epistasis and that's genotyped by environment. But both of them really are genotype by growth rate. It's how fast you grow. And in the case of transporter, I think it actually makes some sense. In that, if you change kind of from a low rate of transport for a necessary nutrient to a higher rate of transport for a necessary nutrient, you can only really take advantage of being able to bring that nutrient in faster if your genome is otherwise capable of doing it. And in the case of something like cobalt, cobalt goes to make vitamin B12. Where does vitamin B12 go? It doesn't really go anywhere. It gets diluted by growth. Right? The only reason why the concentration of B12 will tend to go down is because you're growing. The faster you grow, the faster you need to be able to bring in cobalt to keep up with it. And so, it's the proportional advantage of being able to make use of faster cobalt transport is going to scale with, well, how fast are you growing? How much is it actually even limiting what it is that you can do? And so I think the same is probably true with their transport scenario. And so maybe, and so far there's only been a few examples like this, transport might represent a phenomenon that kind of goes counter to what many other alleles, many other kinds of physiology do. Does that make sense? So I know I just told you one thing. Things tend to be diminishing returns, but I want to make sure that that's a typical sort of interaction. You can have flatliners that are absolutely proportional effect across everything, and you can have upslopers where it actually is synergistic between something generic like growth rate and fitness. All these things are possible, but the diminishing returns have certainly been the most common. This has been seen in yeast work. This is work from Michael Desai's lab, where they introduced, they saw 3 out of 4 tested mutations with diminishing returns. And so I think now, I mean, there's been a fairly large body of papers that have been able to get at exactly this sort of thing. Yes.
Also work with growth rate modulation as your background, like the G by E for diminishing.
We haven't played that game. It would be fun to try. That's a good question.
The synergistic one, where the G by E is similar to the synergistic.
Yeah, it's such a stupidly easy experiment. We should, yes. I will try to write that down. That's a great idea, right? To take the earlier work. We've done other stuff, which has frankly been much more difficult. That would have been a very smart idea to just try playing that game.
And for what it's
Yeah, good.
worth, with the little bit of data that we have with single mutation, it doesn't work.
Interesting, yeah.
But my worry is, I mean, conceptually, I'm not sure that I would expect it to necessarily work in the same way as diminishing because it becomes a mechanism. Right? So
Yeah.
So with the transporter, it makes sense that growth rate should be a generic.
Yeah.
variable.
Well, and I think it's so with the E. coli data, our data, the yeast data, the general pattern has been that a lot of mutations show this effect. Some do not show it at all. So it is not generic. It's not to say every selective effect will go down with fitness, but a surprisingly large number. But they're different. So it's certainly like whatever null model you want, alleles behave differently. That some do fit one pattern and some do not, and some can even have kind of a counter pattern, or be, you still can have kind of idiosyncratic interactions between different genes, and that I'm not ruling that out, but certainly in terms of this generic interaction with growth rate, it's been surprising how common that's been. So let me see what we're doing on time. The time didn't even start, so I guess I still have an hour and a half. Is that right? No, so what might be causing diminishing returns? And in our case, we have at least a guess about this. So it turned out, it's hard to get much morphological details on bacteria. Those are what wild type, look like. They just kind of look like stubby little hot dogs or something, right? There's not a lot to go on normally. And in this case, in wild type, only 2% look kind of different than that. But for our engineered strain, the ancestor with new pathway, (25% of the cells look strange. And what do we mean by strange? They're either significantly elongated, they're kind of hooked like a boomerang, or triply branched. That is kind of like three cells that can't let go of each other. And so those look pretty weird. And we thought, well, that's kind of unusual. What's going on here? And it turns out, and I'm not going to get into all the bar charts to save time, but I'll say that expression of the foreign pathway causes cellular abnormalities. We can make it happen in wild type. We can take it away from our engineered strain, et cetera. And so this seems to represent some sort of cost. And often, for people who try to overexpress proteins in E. coli because they want to purify them, et cetera, the very common sort of phenotype you get actually is elongated, messed up looking cells. Overexpression of proteins often ends up causing some to interact with membranes, interacting with cell division, et cetera. So it also looked like a protein overexpression cost. Yes, yeah. There is the evolved strain. They look great. So yes, it goes away. And so this was the key thing also, is they start off, and interestingly, it's not all the cells. I can get to that later, perhaps offline. But yes, this evolves away. And it turns out we noticed an interesting correlation that lowering the number of defective cells and diminishing returns for fitness. where there was one allele, it was in blue before, PNTAB has no effect on, they grow faster, but they still look messed up just as frequently. And this is the one allele that showed no diminishing returns. It's a 10% effect across all. The other three alleles individually lower the number of defective-looking cells, and they all are the ones that show diminishing returns. Well, that's interesting. Maybe that there's something to be gained from this. And so we developed, so let's ignore the charts for just a second. Oh, I thought I deleted that. So we imagined instead of fitness as just being a single phenotype, we got really sophisticated and thought of it as being two phenotypes. Everything else and protein costs. So it's not highly sophisticated, but kind of growth without costs minus costs. All right? And we imagine that each beneficial allele could affect one or both of those traits. By factors lambda or theta, right? And so the PNTAB allele seems to make you grow faster, the first trait, but the costs seem the same. You still seem to have the same proportion of messed up cells as normal, but the other ones reduce the messed up cells. They also make you grow faster, and so we could think about apportioning this. So you could think of it in this way, that let's say you've got like the ancestral strain. We know that its fitness is 1, but we're saying that we're going to apportion that into 1.14 minus 0.14, because we separately were able to measure how much the costs were. So what we're saying is, if you somehow could have that strain and take away the protein expression costs, it could grow 14% faster. It's just, unfortunately, those costs are there, and so it only grows as well as it does. And so now we're imagining the fitness of any given mutant is going to be some factor that might change the growth rate. So if it's beneficial, lambda will be greater than 1 for that trait, times, we're going to call this growth without costs, benefit and cost there. Or it might also have an effect on protein expression costs, this 14% effect. Then what do we do if we have, say, a double mutant? And our thought was this. So this is just, the proposed model. Would be, if it was a double mutant, let's imagine that there was no epistasis within each trait. That if, like what we were saying before, if you grow faster, one makes you grow faster by 10%, one affects growth rate by 20%. On growth rate, you multiply the 1.1 times the 1.2, and you get 1.32 times kind of growth rate without costs. But the same thing is true for reducing costs. So think about this, right? Like if the costs, like I'm calling there were 14%, if two mutations individually reduce expression costs by half, but in different independent ways, the first one would be a 7% benefit, but the second one, it's not another 7% benefit, it's only a 3.5% benefit, because you've only got half of the cost left, right? That will inherently lead to a diminishing returns sort of scenario. And so we could fit the parameters from wild type and the singles and we assumed that this theta was the proportional reduction in the strange cells. So we use that phenotype. So like these strains, these single mutants, each reduced the total number of messed up cells by about half. And so we went ahead and just used that as a visual proxy. Let's just assume that it's, that the costs are linearly proportional to the number of cells that look messed up, and see how that does. Does that make sense? And so what we wanted to ask is, if we do this, could we now predict the double, triple, quadruple strains. And indeed, that's what I'm showing up here. So for each one of, well, that's fine, I guess, as it was. Each one of these charts, the one with noise is the data, and then the triangles are the model. And so what you can see, like for P and TAV, the data are flatline, the model's flatline. Like look at GSHA, we were able to predict actually pretty darn well the fitness effects, where we've just gone from fitness as one black box to I'm going to pull one thing out of that black box that we think we can measure, and then the rest of it is still just a black box. But even just partitioning it into two phenotypes, the model had an R squared of 0.97 as two phenotypes, whereas the simple one phenotype, just take fitness and multiply them together, was significantly lower. Now, obviously this exact phenomenon, cost reduction, I think if there may be aspects of like the E. coli data that have to do with cost reduction, but this could, it's not to say that this is going to explain the yeast data, that data, but I think this represents one of multiple types of physiologies where if you understand kind of what's going on, you can develop a different null model that does have kind of inherently built in that you would have diminishing returns. That if you're reducing a cost and you have independently acting mutations, you should see kind of a diminishing return epistasis emerge. Does that make sense? All right, so diminishing returns, diminishing returns, diminishing returns. Then a new paper came out just from Michael Desai's lab in 2019. And at first it looks very similar. So what they did here is they took various yeast strains that had high fitness and they introduced deleterious alleles. And because in yeast it's easy to do these sorts of matings and then use sequencing and barcoding to their advantage, they did a ton of this. And so they had this nice big data where they introduced deleterious mutations into these fit alleles. And there was a generic scaling with background fitness. Oh, okay, so it's the same kind of thing. Why not, right? Like, if we saw it in one way, why don't we see it in the other? I can get So there was a generic trend with fitness. The mutations had the biggest effect on high fitness backgrounds. So at first it seemed like this is just the same song being rewritten with slightly different words. And then you get this really uncomfortable feeling that you realize, wait a moment, this is exactly backwards to everything I just showed you. For beneficial alleles, when combined, in general, across many different systems, they tend to be less and less effective. That, like if you were to march backwards, if you were to take the fully fit strain and march backwards from the 11111 to the 00000, as you take away the beneficial alleles, they would have been less deleterious if you remove them first than if you remove them last. Here, they're actually most deleterious on the fit backgrounds. And so it is exactly the opposite trend. What the hell does that mean? But I think this is where we have to recognize these represent very different classes of mutations. When we combine successful winning beneficial mutations, they are a really rare class of all the possible changes that could occur. And they, remarkably, across systems, have properties that are at least highly common. Not universal, but highly common. Interestingly, deleterious alleles also have properties which are quite common. Not all of the alleles showed this, but many of them did. But it's the opposite trend. So how to put this together into kind of a thought, A friend of mine at the University of Idaho wrote the perspective piece for this paper in Science, and we spent a lot of time talking about this, and he came up, I think, with a really fun fitness landscape that now the X and Y axes, they both represent genetic space, but he's kind of like partitioned the types of mutations in different directions. So marching along, like the ridge is this diminishing returns sort of thought in red, that as you stack more and more mutations, you tend to kind of see less and less and less proportional effect. Adaptation tends to slow down, and we all feel kind of pretty comfortable with that. And independent from that, this idea that, well, if you hurt, if you pop the tire on a Porsche, the drop in speed is going to be higher than if you pop the tire on, some little rickshaw or something, right? Like, okay, that feels okay too. But when you realize that you can't have it both ways, that it suggests, at least for these different types of mutations, kind of a different form of kind of typical curvature. It cannot, so first of all, that there is curvature, but they're different, right? You cannot have one null model that explains both, right? And so there are these interesting trends with fitness, but the type of change that occurs can very much affect which type of trend you see. Yeah. Well, I think your intuition is correct that there will be many more deleterious mutations possible than beneficial, especially in these lines that are already starting to adapt and improve. I don't know that you need to confirm that one class is more numerous than the other for this trend to still be kind of true and be interesting. And I don't know if it's necessarily just the rarity of one versus the other, because you could pick any trait and say, all right, well, this is a rare trait, but it doesn't mean it's necessarily going to have a different pattern of epistasis. So I would say at this point, this presents a big puzzle. I don't think anybody has a single synthetic picture. Certainly the authors didn't, neither did, besides like pointing out, shit, these are two different things. Craig and myself, like, it's not clear where, but I think we should, I think it's easy to end up feeling very comfortable that, oh yeah, sure, things are going to slow down as you improve in fitness. Or feel very comfortable, oh yeah, high fitness strains are going to really be hurt, but to realize that they're two sides of the same coin, and suddenly very uncomfortable in terms of thinking about, well, why? And I would say we don't know. So you guys should study this and come back with an answer in 2 years. I think we just really don't have a good picture of this.
I have some trying to get my head around it. Are you saying that fitness gain is in large steps, whereas small steps that fitness declines are large steps?
No, I'm saying, well, no, because this isn't the DFE. This isn't the average size of what's possible. This is saying that the magnitude of an effect of a mutation scales generically with growth rate, but that for the beneficial alleles when tested, they tend to have more proportional effect on a low fitness strain than a high fitness strain. Whereas for deleterious alleles, they tend to have more of effect on a high fitness strain than on a low fitness strain. Which, it's interesting. I mean, anything is beneficial or deleterious just based upon kind of like the order in which it happened, right? Like if you started with a deletion allele and you add the gene, now it's a beneficial allele, right? Or if you started with one allele and just flipped to the other allele status, right? Like adaptation is in the eyes of like what actually happened kind of historically.
It matters where on the ridge you are.
But these are the same, but they're the same starting strains. So like the strains that were being tested, in this case, the yeast, were exactly the same strains that they were doing mutational combinations with and saw one trend, and now put in deleterious mutations and see an opposite trend. So they're starting even at the same spot. They're literally the same strains. So it's, like I said, I'd love to have even a decent hypothesis as to why. And right now, I don't have that. right? I think that's the state of our knowledge. We just really don't have much clue. All right. I guess I can quickly mention, and then I'll finish off. Yeah, I'm almost there. All right. So let me just quickly mention a couple related aspects. And if anything, this could be just directions for future work, for your future exploration. I don't want to explain all this in great detail. Given the shape of fitness landscapes, then, particularly in cases where there might be either multiple ridges or multiple peaks, then there becomes a question kind of how can you access kind of other peaks if you're kind of locally close to one, right? If your job is to just simply take steps, but only take steps that are uphill, it depends upon where you're set down on Google Maps, and you can find, some equal path to whatever the nearest thing is, but how would you get necessarily somewhere else? And so there have been a number of nice papers. This is one that came out this last year from Andreas Wagner's lab, looking at fluorescent proteins, where they basically had the idea, what if we just kind of spread out the population first kind of neutrally for kind of one trait. So you start with different initial genotypes and now ask for a new trait to evolve. And so they used YFP. They first went through a few rounds of kind of like directed mutation, but kind of in vivo selection of YFP, spread out YFP variants with purifying selection to be yellow, and then suddenly switch the selection on the flow cytometer, the FACS sorter, to green. And yellow fluorescent protein's a little bit green, but not very. And then ask kind of how quickly could you evolve green fluorescent proteins if we just start with the original genotype versus if we first kind of spread it out. And from this sort of work, they found, this is going to look a lot like a poster that you can go and see with these nice little cubes. You can draw a cube. Hypercubes, just flatten them out. Like they just look awful. But what they found is this actually was a fairly effective way to cross fitness values, that when they reconstructed some of these strains that had, say, a triple mutation, A, B, C, that if they had only selected for green fluorescence, every single path to ABC would be blocked by deleterious steps. But kind of the neat thing was, when they reconstruct them and test them in yellow, they were neutral. They could just kind of neutrally wander around. And this is, quote, cryptic variation is yet another word that's used for this. And from some of those, like the AB genotype, as long as you neutrally get over to the AB genotype while the world is selecting for yellow, now when you move to a world selecting for green, it's simply an uphill climb to get from there, mutation C, and you're bright green. Right? So it relates to kind of these same sort of concepts. For the sake of time, I'm not going to hit too much more there, but there's a lot of fun directions about accessibility. Like I said, there's a neat poster here at the conference in this regard. And this is an example of kind of these mutations. Not surprisingly, given that there were protein mutations, it depended on kind of exactly what background they occurred upon. And not only this is evolution that happened in the lab in just a few rounds of selection. This sort of stuff has happened over time, in the past. And I'll just point out some neat work in case you're interested in this. Joe Thornton, who was at Oregon and is now at the University of Chicago, has used as a model system hormone receptors in the vertebrate lineage and has done phylogenetic reconstruction. So when you make a phylogenetic tree, you also have guesses for what kind of deep nodes, like some ancestor of skates versus bony fish and vertebrates. And if your guess is really good, you can now call up a company and say, all right, synthesize this gene for me, and you can test properties of it. And if you were good enough to actually make a gene that can still fold and things like that, you can ask questions about, well, what was its phenotype? And so, very long story short, there's a lot of neat papers in this direction. From this phylogenetic reconstruction, they test these old nodes. They've found like relatively few mutations being required to change the specificity of these receptors for different hormones. And the neat thing is that it relates a lot to the poster that you could see, that there were neutral mutations for specificity that are required for the functional changes. so that the very mutations that actually allow the receptor to change what it is that it binds to, they end up destabilizing the protein. And so you need these other mutations that stabilize those exact contacts, but the stabilizing mutation alone doesn't do anything when it comes to hormone receptor capacity. So like the poster, this is something where you've got historical contingency. You had to have actually evolved those changes first. There's this neat paper where they tried to use selection in yeast to rerun the tape, and they got back the historical mutation and none others. And so it actually does kind of sound similar to Dan Weinreich's main story that perhaps at least the evolution to a particular outcome might be highly constrained by epistasis. Now, it's not to say that's the only outcome that could have occurred. Perhaps it could have fanned out, but at least the one that did occur may have been bottlenecked through relatively few events that needed to have happened in the past. All right, it's a fun story out there. It's a neat system for people who might be interested. And so these were, like I said, these were very specific mutations, very rare. Is there no hope for protein biologists that there might be some generic trends? And there is actually. There's Nobel Prize-worthy trends. Frances Arnold, in a lot of her work in terms of directed evolution of proteins, one of the huge things that they've found is kind of a generic trend that can kind of orient one's thought is thinking about protein stability. What's the delta-delta G of folding? And there is a really important trend with proteins that if you look at their stability versus the percent of the protein that folds, this tends to be highly cooperative. That over a relatively narrow range, you go from most of the time the protein can't fold at all, or if it does, it'll fall back apart, to just a little bit more stability, they'll stay folded. Proteins are really only marginally stable. And so where does that come into function? That's just folding. It turns out both stability and activity are both rare traits. So if you just put random mutations into proteins, the vast majority of those will kill one of those traits or the other or both. And so if you select for function, chances are, just statistically, it's not a necessary trade-off, but statistically, most function-improving mutations to proteins will also cause destabilization. So if you were only marginally stable, and now, hey, I've got a mutation that allows me to use a new substrate or something, but it pulls me to being an unstable protein, I don't get to take advantage of that. So they had the basic idea that then, if you start with a very modestly stable protein, and there's kind of like the probability of where new proteins could arise in the stability versus activity space, there'd be a very little corner of things that would actually stay stable and actually be more active. If you simply bump over, kind of like shown there with that MG to TG mutation, if you give yourself a little more space, just first make the protein more stable, now select for function. Because then you have a much larger space of possible function-acquiring mutations that won't absolutely mess up the protein. And indeed, this has worked really well, and has led to a very nice career. And a lot of practical outcomes have come from this. All right. That's the end of day one. I knew I was optimistic anyways. Before we move to evolution of metabolism, which I will go ahead and get started here, and then we'll pick up again in the later session today, any more questions or thoughts about epistasis? Now, I'd certainly be happy to chat, whenever otherwise. Yeah, I think that should work just fine. You could have moved in natural alleles or do matings, and yeah, I mean, on one hand, it's kind of obvious. Like it's hard to imagine it not working. That more genetic variation to start selection would probably be better than not having it. So it's not. There are subtleties where I think do begin to get interesting and play into kind of where we were seeing epistasis can have kind of different sorts of trends. I mean, the interesting thing there is to explore space without damaging the protein's function. Like you could call that genetic robustness, for example, that you can tolerate many mutations and still do what you're doing. But if you want to acquire a new phenotype, you better not be robust. You want to actually get some damn change from the mutations that occur. And so robustness, thinking of it as like a single capital R holy term, like it ends up like kind of working both for and against you for this kind of same kind of property. And so I think it can lead to kind of confusing and sometimes disparate views, worldviews on these sorts of things. And I think it becomes really important to think about kind of genetic robustness for one trait versus genetic robustness for another trait, or environmental robustness, et cetera. And if those things are too lockstep, then again, like you could maybe spread out, but you can't change phenotype, or you can easily change phenotype, but you also can't keep your phenotype. And so there's definitely kind of pros and cons, so as to say. Yeah. All right. Yes.
So on average, I mean, to take a sample of proteins, like where does one find that average on that plot, near the station or far away on the
They tend to be not right at, but like just a few kcal per mole, negative to maybe like 8 or 10 kcal per mole negative. And just to give a sense of scale, like if I remember correctly, that's even a fairly stable protein. It's about 1/3 of the energy of breaking a single ATP would unfold a protein. So they are not, like the amount of energy there, like it's actually remarkably small. Proteins are, tend not to have a tremendous amount of stability compared to what would cause them to unfold. Perhaps, but they needed to have folded in the first place. And so perhaps to have an effective folding pathway from nascent growing chain to becoming, because you can certainly design proteins where you can introduce salt bridges and this and that, or cysteine crosslinks and things like that. You could design proteins that are much more stable, but they don't tend to be very many like that. I think that's a fair statement. I'm not a protein biochemistry expert, but I think that's about right. Okay. So, let me put this down. Evolution of, where did I go? Evolution of metabolism. So I'm going to start with a question for you guys. Imagine you have a pathway. It's got 4 enzymes. It's going from a substrate to a product. And this pathway comes to exist in a steady state. And I'm guessing for this crowd, at least for the majority, I probably don't need to over-define what a steady state is, right? All the fluxes, all the concentrations are unchanging through time. Another way of stating it is the flow into any one of those metabolites better be the same as the flow out, otherwise you're not in steady state. Okay, so you're at steady state, everything's hunky-dory. Now, imagine you have a mutation that would double, say, one of them, enzyme B. Okay, what's going to happen? What will happen to the system in terms of a few different traits? And I do want votes for this. No abstentions, hopefully. What about flux through the pathway? If I pick an enzyme, in random, I'm not telling you anything about the enzyme properties, et cetera. Is the flux going to go up, stay the same, go down? And what about the different metabolites? The one that's upstream of it, Xi, or the 2 metabolites downstream, Xj and Xk? Are they going to change, going up, stay the same, or go down? Make sense? So why don't you take like a couple minutes to think about this, what you would expect to see, and while you're beginning to cogitate about that, like my motivation for talking about the evolution of metabolism partly is because I think like metabolism itself is important. It's worth talking about in and of itself. But it represents somewhere, as I think you'll see, where we have some ideas, if we want to link from genotype to phenotype to fitness, ultimately there's actually even more steps there, right? Because a genotype is going to affect you can get really nitty-gritty. It's going to affect like transcription, and then RNA stability, and then translation, and then protein folding, and then protein activity, and then right? And some folks like Deepa study many of those first sets of steps, et cetera. And this way of viewing the world is a way of getting from, if I can at least get to like the biochemical phenotypes, kind of how much of enzymes and what are their properties, what can I say about growth, which at least in these circumstances I'm going to talk about, is essentially fitness. And so it's a way of getting from kind of one level of this genotype to phenotype map out to something else. And as you'll see, it can actually be pretty useful. Okay, oh, yeah, I do it later, but you're right. This slide was from a class where that had already happened. Thank you very much. This is the activity per molecule of an enzyme. And usually it's in something like per second. How many reactions per second? This can do 10 per second, or it can do 20. And actually, you could double the enzyme's expression level. And because I said before, they're multiplicative, it would actually have the exact same effect. But let's just think about it as that. All right. Yes? Oh, yeah. It does not matter what the concentration of S is for this question. However, there is a detail that I was going to get to in just a minute, but I was worried it'd be a little too complicated for here. Imagine like that as being kind of like this, like the pathway. S and P are kind of external to the system and held constant. So whatever the concentrations are, imagine, like substrate in the external world that effectively, if I want to define growth in the next minute, there's no real change in the glucose concentration. And there's no real increase in bicarbonate or acetate or anything that gets spit out. So imagine those kind of being outside and everything else inside. All right. So, ready to vote? What do you think about flux? If we double an enzyme, is flux going to go up, stay the same, or down? Try to keep to your vote. So who says same? I think I heard a same. We've got some robustness fans. I'm going to call that about 7-ish. All right. Who thinks it's going to go up? We've got some people who are a little bit more optimistic, maybe about 10. Down? All right, nobody's feeling too grumpy. Maybe you had your coffee or chai. All right. What about the concentrations? Will they change? So what about XI? Same? We've got kind of one down. 1, 2, 3, 4. Up? All right. What about the ones downstream? XJ, immediately downstream? Same? No sames. Up? Down? All right. What about XK? It's now a couple steps downstream. Same? Up? Down? All right. Well, these represent interesting vote totals. I noticed that the sum of the votes is not entirely constant across these different scenarios. And in no case does it represent the entirety of the classroom, but okay. You can see clearly it's possible to have some kind of different views on what this would look like. So I'm going to provide just a little bit of background. You guys probably didn't think you were going to hear this or that it would have anything to do with evolution. I'm going to hopefully get to the point of you feeling like maybe it does. Biochemistry. I teach that and I've taught evolution before. So I get to torture you with both now. One substrate, irreversible Michaelis-Menten kinetics. Basically, how does an enzyme work with respect to substrate concentration? And so if we have, starting simple, this is just a product going to a substrate catalyzed by an enzyme. And we're going to leave the enzyme level constant, but we're going to vary the substrate concentration. What type of shape would there be for a relationship? As I dial the substrate up, is it going to go up linearly, less than linearly, more than linearly, hill function, sinusoidal, pick something. You like functions, right, Lindy? Like what function would you like to throw in here? Did I? Oh, you are absolutely right. I need more coffee. That is certainly true. Substrate to product, yeah. Yeah. It still would be irreversible, but it would be backwards. But yeah, this would be a little easier. Was that a sinusoidal? So it is sinusoidal if you plot out on a log scale. And so this isn't a log scale, this is a linear scale. Okay, all right, yeah. So this has this sort of a shape, right? Anybody know offhand what the math would be? I can't call out you physicists, I could call you biochemists, but I might not get a single answer. All right, so this sort of shape, you can characterize it by a couple traits. One is Vmax, where as the enzyme just cannot work any faster. It's no longer having a hard time finding substrate. There's no particular waiting time for the substrate to get to the enzyme. It just can't work any faster. That is, given the amount of enzyme that's there and how active it is. Now, Supreet, I know that you know the answers for these questions. You're just staying silent for all of it. Okay. Man, bad. So, Vmax is, the maximum capacity at high S. And ultimately, this flattens out. At very low concentrations of substrate, there's plenty of enzymes. Substrate's very low. And so it's more the waiting time to find substrate. And as substrate concentration goes up, for a little bit, it's going to increase linearly at very low S, but it has this kind of curvature. And we can write this out. The velocity of this reaction will be Vmax. And then somehow substrate plays a role, and it plays a declining role. And the Michaelis-Menten approximation, which has to abide by these various assumptions, states that it looks something like this, right? So you get this kind of hyperbolic shape. And that Vmax term is easy to understand, but there's this little extra term, Km. This is the Michaelis constant. So what if S was equal to Km? So Km, interestingly, Vmax is in units of activity, and then this ends up being dimensionless because it's concentrations over concentrations. So that would be like millimolar per second in terms of Vmax. Km is actually a concentration. So imagine S equals Km. Where would I be on the graph? Half, right? Because you'd have 1 of whatever that number is over 2 of whatever that number is. So Km is just telling us kind of where the halfway point is. It is Vmax over 2. It's just right there, right? And so you can then define a whole family of these curves by, well, what's the Vmax, what's the Km for these? And yes, this would be in millimolar, and Vmax would be something like millimolar per second. All right, great. But that's if there's just one direction. And life, that's too simple. You guys are up for more than that. Most reactions, vast majority of them, have some potential to go both ways. I'll try to get my S's and P's in the right direction. We have this. Okay. So what's that going to look like? That, you just need to write this out, is 2 half reactions. The Km for S minus Vmax reverse P and the P plus the Km for P. All right? And so this sets up a tension between backwards and forwards. And so if you plot this out, this is the overall rate, the net rate of the reaction, where positive would mean that it's going forward. Negative values would be reverse. 0, if the net flux is 0, then we'd also describe that as equilibrium, that you have exactly the same amount going one way as it's going the other way, right? And so you can look at this space and you can find, for example, like maybe a line of equilibrium, that all along that line is where the forward and the reverse reactions are the same. And thus, if we kind of project down from that line, there'd be a space of, depending on what the concentrations were, all of that if the product is that high and the substrate's that low, all of that is going to be where the reaction will flow backwards, and thus all of that space is where it flows forwards. Make sense? Fairly straightforward. So I'm going to rewrite this, remembering that Vmax is simply the enzyme concentration, how many there are per cell, times kcat, the specific activity of each enzyme, which is in per second. All right? Because the important thing is, if you change the enzyme, and this is getting to one of the main points that's going to come up later, it's going to actually change both of these, right? Things will flow forward faster, but they'll actually flow backwards faster also. And so if we factor out the enzyme, now we just have, kcat forward times all of that minus kcat reverse. Make sense? This ends up being rather important for what we're going to end up seeing later. All right. 1101. All right. 3, 4. Let me ask you one more question, and then I'll let you out for... life is also not so simple. It can go both directions, and you can have more than one substrate. If you do, then you just got more than one saturation. You could be waiting around for one, you could be waiting around for the other, you could kind of be waiting around for both, if they're both low, etc. And they both have to be high for you to be able to go at full speed, right? And so you just, in terms of this approximation, you can just multiply them out. This can get much nastier if you want to get into particular enzyme kinetics and think, God, I'm not going to go that direction. So we're just going to leave it like this. But this also becomes important later. So I'm going to leave you with this thought that relates to kind of the first. Let's imagine, I'm going to tell you the Vmax for each of these enzymes. All right? If I were to ask you, if you've had a biochemistry class, and you may not have, what sets the pace of metabolism in a cell or in a pathway, the very common answer is on the title, a rate-limiting enzyme, which seems like a very straightforward concept, right? Which one of these would be the rate-limiting enzyme? B, right? It looks pretty obvious. B is slower than the other enzymes, right? Right, and the rationale, the slowest holds up flux for whole pathway. All right, and what this would look like then visually is, so now we're going to have S constant. And what if we just change levels of enzyme B? Where were we? We were at a concentration of 1. Remember, they were 3, 1, 5, and 7. So if it has a Vmax of 1, so let's actually, so that's your flux right now. What would happen if I were to make it go higher? What if it was 0.9? How fast could this pathway work? 0.9. If it was 0.1, it could work at 0.1. What if it was 2? Huh? Well, by the idea of a rate-limiting enzyme, if I had told you, imagine I put a 2 there instead of a 1, you still would have called that the rate-limiting enzyme. So if it's 2, okay, maybe you could work up to 2. But 2.9, maybe 2.9. What about 4? Now you would get capped off at 3, right? And so this idea of a rate-limiting enzyme would suggest this picture, that wherever the next lowest is, which in this case happens to be 3, you could go faster until you hit that wall, and then that's now, at least in terms of enzyme B changing, like that's as good as you're going to get, right? And so I'm going to spend my next section beating this concept out of your head. How this actually is entirely the wrong scenario to be thinking about. Here, it can exist, Now, this may not be true of an Indian road because we Americans are maybe a little bit more behaved when it comes to what to do when there's other cars in front of you. But at least, normally for us, on a 2-lane road, if some jerk in front of you is driving slowly, you're probably going to be stuck for a while until there's a passing area, which I think is basically the entire geography of the country here, is where you can pass. Or like this bucket brigade, right? If you're passing things along subsequently, whoever's the slowest person passing these buckets to put out a fire is going to hold up the line for the rest. And in those scenarios, this intuition is absolutely true. People have used this idea to think about ribosomes moving down a transcript, right? Because you can't get by, right? Like you're just going to be held up. All I need to know is how fast you can work, and whoever's the slowest is going to hold up the rest. And I'm going to try to explain to you, based upon the biochemical kinetics, and ultimately related back to selection, why that does not hold for metabolism. The picture is substantially more nuanced. I'll do that, and we will get into using our, whenever it is in the afternoon that I've got the tutorial session. All right, I think we should caffeinate.